Okay, so good morning and thank you for listening to the presentation. It is my pleasure to give you an update on the CATS activities. Because we don't have that much time for presenting, I thought to quickly go through our work plan and highlighting the activities that might be of most interest for these working parties, and then also to briefly stop on the most recent approved ATMPs. Next slide, please. So just a few words on how we work and why the work at the committee makes it very different to the rest. What CAT stands for is safe and efficacious first-in-class ATMPs. We also stand for including incremental scientific and clinical knowledge in regulatory decision-making. We support ATMP developers, and we are happy and proud to incorporate physician and patient perspective in our deliverables. We support patient access by addressing root causes of real-world data deficiency and also by linking with HTA bodies. We warn against unproven cell-based therapies, and we strengthen communication and exchange with EMA committees and working parties. Next slide, please. The main differences between ATMPs and other medicines are basically that they are complex products to develop, manufacture, characterize, and test. Therefore, they follow non-standard development programs. They frequently develop novel toxicities. Therefore, there has to be a risk assessment on the insertional mutagenesis events. They must follow specific post-authorization obligations because frequently after the trials and the assessment process has been done, there are remaining uncertainties. And it is very frequently that there are no precedent cases for regulatory decision-making. Next slide, please. So going to our 2023 work plan, the activities that I wanted to highlight are the following. First, on enhancing the benefit risk methodology. What we're looking is, of course, balancing benefits and risks in a way that they are robust, consistent, and as transparent as possible. And for that, we are contribution to the finalization of the reflection paper on single-arm trials. Secondly, on our efforts to improve interactions with HTAs, we are holding product-specific discussions on newly approved ATMPs by setting up meetings and webinars to present to HTA bodies the scientific grounds of the approval. Thirdly, on the post-authorization safety and efficacy follow-up of ATMPs, the CAT will prepare this year a scientific publication on the follow-up of patients treated with AAV-based gene therapies and patients treated with CAR T cells. And finally, on the real-world data side, the CAT is keen to use enhanced analysis of real-world data to complement the and give an additional perspective to the evidence that has been obtained from clinical trials. And there is one specific activity that is a project that I want to present to you very briefly that we are finalizing this year, which is the first EMA-funded pilot study on SMA. Next slide, please. So this first pilot study has used spinal muscular atrophy as the disease that could be ideal to pilot this exercise. So SMA is a monogenic disease that in 4 years of time has gone from not having any therapeutic option to have 3 approved therapies, being one of them gene therapy. And therefore, it could be like the good disease to try to understand how real-world data could help to follow up in a post-approval environment. So what we are doing is we are looking at information containing already existing registries. Sorry, next slide, please. So we are looking at the information contained in existing registries with the objective of describing the natural history of SMA, the evolution of the care management over time, and the disease progression considering the new DMTs. using, as I say, the data from registries. A very important secondary objective is that we want, with this pilot, to evidence the challenges that these looking into the information that the registries have gives to us, and the lessons learned by performing this study. We know that the real-world evidence is getting more and more attention from the EMA, and it is a great exercise to understand the challenges that we're going to be facing on a real case. Next slide. So the design is, of course, a non-interventional descriptive registry-based cohort study of SMA patients. The source will be 7 European registries covering 10 European countries, in which there will be some that are clinical reported, some are patient reported, some are mixed, and something that I find it has great added value, some of them have patient reported outcomes, so for specific areas that are relevant to patients. The data will cover a period from 2010 to 2022, and we will be studying the data of some 2,000 to 1,400 patients. Next slide, please. I'll jump that one in the interest of time. So next slide, please. I won't dedicate more time to that study so that I can have some time to speak about two of the most recently approved ATMPs, in which, of course, the CAT has had some input. First of them is a gene therapy for AADC deficiency, which is a severe neurological condition. It's an ultra-rare disease that we know about 200 patients. It consists basically on the loss of dopamine production and leads to a very debilitating situation. The indication of Upstaza, this gene therapy, is for patients aged 12 months and older with a confirmation of the diagnosis and with a severe phenotype. The therapy consists on a gene replacement based on AAV2's vector, and the method of administration is through an injection directly to the brain. Next slide, please. Some of the aspects that have been specifically discussed at the CAT were the comparability of the manufacturing processes, the relevance of the data that came from third countries, the specificities of the administration procedure, the extrapolation to older or younger patient population, the non-comprehensiveness of data, and the granting of marketing authorization under exceptional circumstances. And then, of course, the follow-up data with the registry-based studies. Next slide, please. So the other gene therapy that has been recently approved, which was in July 2022, is another gene therapy, Roctavian, to treat severe hemophilia A. This went through a single-arm trial that lasted for 2 years and involved 138 male adult patients. There were some other therapeutic options, but they were burdensome. They implied weekly or monthly administration. And with this single intravenous infusion, the results obtained have been very positive. Next slide, please. So after these 2 years of study, 75.4% of the patients had decreased their symptoms to those of mild hemophilia. The yearly number of bleeding episodes decreased by 85.5%, and the need for additional factor VIII replacement treatment dropped by 97.5%. The safety concerns are mainly around the hepatotoxicity treated with corticosteroids. And then, of course, there are needs to long-term follow-up for 15 years. And that's all I had the time to say. So next slide. Thank you very much.